Hello, my name is Paola Vermeer, and I'll be taking you through my poster that's titled Tumor Innervation and High-Grade Serous Ovarian Carcinoma. My lab has been studying tumor innervation, and we became interested in ovarian cancer given the poor prognosis that these patients have. We know from other studies that patients with densely innervated tumors do poorly, and so we wondered whether the poor prognosis of ovarian cancer patients may be contributed by the extent of innervation in their tumors, which had not been previously studied. Ovarian cancers can be divided into several subtypes. High-grade serous ovarian carcinoma accounts for the majority of cases and the majority of the deaths. There's also low-grade serous, clear cell, endometrioid, and mucinous. So we started with just an immunohistochemical analysis of these subtypes of ovarian cancer, where we stain them for beta-3 tubulin, which is a neuronal marker, to define whether how these different subtypes are innervated. And here are just some examples. And what we found was that high-grade serous ovarian carcinomas are really the only subtype that are densely innervated. The other subtypes were really very lowly innervated or actually lacked intratumoral nerves altogether. We had previously published that tumors release small extracellular vesicles called exosomes that lure local regional nerves to the tumor bed. So we wondered whether this difference in innervation that we saw in the patient samples was also reflected in the neurite outgrowth activity of their exosomes. So we purify exosomes using a differential ultracentrifugation technique, which is outlined here, and that we've previously validated a number of different methods by Western blot, electron microscopy, and by fractionation, and show that this method really does do a really good job of purifying exosomes that have neurite outgrowth activity. So we purified exosomes from human ovarian cancer cell lines that represent the different molecular subtypes of ovarian cancer and tested their neurite outgrowth activity in vitro in this assay here. So what we do is we isolate dorsal root ganglia from mice, and we can culture these DRG, and then we stimulate them with 3 micrograms of exosomes, and then allow the exosomes to induce neurite outgrowth for 48 to 72 hours, and then we analyze that by Scholl analysis. In all these DRG assays, some DRG are treated with nerve growth factor, NGF, as positive control, or with vehicle alone, which is PBS, as our negative control. So when we tested exosomes that represent the different subtypes of ovarian cancer, we found again that the majority of the high-grade serous ovarian carcinoma exosomes had nice, robust neurite outgrowth activity seen here in the brown. The orange is the neurite outgrowth activity present in NGF-treated exosomes. And what you're looking at here is the quantification of the Scholl analysis, where we can look at the number of intersections, so the complexity or the number of bifurcations that the neurites extend, and the length of the neurites. And obviously the negative control PBS-treated DRG show very little neurite outgrowth activity. So by and large, the high-grade serous exosomes had a high neurite outgrowth activity. Exosomes from clear cell carcinomas had very little, if any, neurite outgrowth activity. This was also true for endometrioid and mucinous. We only had 1 cell line of mucinous, and this one happened to have a little bit of neurite outgrowth activity. As a control, we tested exosomes purified from normal fallopian tube epithelial cells, which are the cell of origin for high-grade serous ovarian carcinoma. And these exosomes also have no neurite outgrowth activity. So this in vitro assay for exosome-mediated neurite outgrowth activity very nicely correlated with the immunohistochemical analysis that we did of the patient samples, again suggesting that only the high-grade serous ovarian carcinoma subtype are highly innervated. Then the question was, what do these intratumoral neurons do once they're at the tumor bed? We know how they get recruited. We know that the high-grade serous get highly innervated, but what are these neurons doing? And so one way to assess whether they're functional or not is to do electrophysiology. And to do this, we use microelectrode arrays, which are these small arrays that you can buy in different configurations. And essentially in the center of this well are several microelectrodes. The ones that we use have 60 microelectrodes. And so we take tumor, this could either be from a patient, a freshly excised tumor, or from a mouse, and we take a slice and put it over the electrodes. And this is connected to an amplifier and a computer, and you can look at your sample under the microscope, and we can then measure the electrical activity coming from that slice, which presumably is coming from neurons present in that slice. We also did proximity ligation assay using pre and postsynaptic markers to determine whether synapses or synapse-like structures are being formed within the tumor bed. And the red puncta that you see here are positive signals for PLA. And we found that there was significantly elevated PLA signals in high-grade serous samples as compared to normal ovary or benign cystadenomas. Again, this is consistent with the intratumoral neurons are forming functional connections at the tumor bed, and MEA is one way that we can measure this activity. So when we did this, and we compared the electrical activity between malignant high-grade serous samples and benign cystadenomas, we found that at baseline, the malignant tumors had significantly elevated activity, and also their evoked activity was also significantly elevated. This is consistent with the highly innervation status that we see in these densely innervated tumors compared to the others. When we quantify this innervation, we decided to look at the impact of BRCA mutations here. 20 or 30% of patients with ovarian cancer harbor BRCA1 and or BRCA2 mutations, and these are associated with poor prognosis. So we wondered whether those cancers that harbor BRCA mutations might have an increased extent of innervation or may be influencing the innervation in their tumors. So when we looked at the high-grade serous ovarian carcinoma samples, segregating them by BRCA status, so those that harbored BRCA1 mutations, or BRCA2 mutations, or were wild type for BRCA, it was really interesting because we found that those tumors that harbored BRCA1 mutations actually led to a decreased electrical activity compared to the BRCA wild types or the BRCA2 mutated. We do have a murine model of high-grade serous in which BRCA2 is deleted using CRISPR-Cas9. And when we look at that tumor, so we implanted the parental tumor, which harbors P53 and P10 mutations, which are very commonly found in cases of high-grade serous, or the addition of a BRCA2 mutation, the BRCA2 mutated tumors had a significantly elevated electrical activity, again, suggesting that BRCA mutations do impact the functionality of these intratumoral neurons, both in the animal and in the human. Finally, to confirm that the electrical activity we were seeing by MEA was actually coming from intratumoral neurons, we use a double transgenic mouse where we crossed the TRPV1 Cree, since we had identified that TRPV1 expressing neurons were infiltrating the tumor. We took the TRPV1 Cree mouse, crossed it with the Rosa DTA, diphtheria toxin mouse, to generate a TRPV1 DTA mouse. So in this mouse, the TRPV1 expressing neurons are genetically deleted. And you can see that here when we isolate DRG from the TRPV1 DTA mouse and immunohistochemically stain for TRPV1, there's none. But when we harvest DRG from control mice, that you can see the presence of TRPV1 expressing neurons in the DRG. When we implant tumors into these transgenic mice or controls and then look at the MEA analysis, we see that at baseline, the TRPV1 DTA tumors had significantly lower electrical activity than the controls, but at the evoked activity was also lower in the TRPV1 DTA tumors, suggesting that at least some portion of this electrical activity that we were measuring was coming from TRPV1 expressing neurons. And the last thing we did was we wanted to see whether we could silence pharmacologically this electrical activity, suggesting that this might be a potential therapeutic avenue for densely innervated tumors. So we took one tumor from case 2557. We measured the MEA electrical activity at baseline, the evoked activity, and the reversion. And then we took those same slices, incubated them with lidocaine, which is a voltage-gated sodium channel blocker, and then measured the electrical activity again. And we saw there was a robust attenuation of all the electrical activity at baseline, evoked, and reversion following treatment with lidocaine, suggesting that this may actually be a clinically useful approach to silence intratumoral neurons in patients. So I hope what we've learned today is that high-grade serous ovarian carcinomas are highly innervated and that the other subtypes are lowly or not at all innervated. High-grade serous ovarian carcinomas also harbor elevated electrical activity. The exosomes purified from high-grade cell lines also have a high neurite outgrowth activity. This is all consistent with resulting in a densely innervated tumor. The electrical activity that we measure largely stems from the TRPV1 expressing intratumoral neurons, and that loss of BRCA2 in the tumor significantly elevates the evoked activity of these tumors. Tumors with high electrical activity may actually be quite sensitive to pharmacological blockade, suggesting this may be a therapeutic option for these patients. I'd like to thank the Dakota Scholars Award in particular for funding this project, and I also obtained funding from the Vassar Center for BRCA at University of Pennsylvania. Thank you.